you in the frame of mind for lecture? No. Bad question. Because you have no choice.
Yep. Okay.
Okay, so we try to go a little bit slower today. Let's see where it leads us. So last lecture we talked about glycolysis. You still remember glycolysis? Right? We talked about how energy utilization, the first step is glycolysis. We looked at how glucose is being broken down to pyruvate. Right? The end of glycolysis is the product pyruvate over here. Right? And we talk about Usain Bolt, who can do like a sprint in 100 meters in 9.58 seconds. And during that period of time, what he essentially is doing is an anaerobic glycolysis, right? Because there's simply no time to break down pyruvate completely to carbon dioxide to water. So what he is doing is really to power himself, get his ATP from here, and then regenerate his pyruvate in his muscles, right, to lactate, right? And then the build-up of lactic acid is what you know as very soon, right? Every time you do a very vigorous exercise, after that your muscles is soon, okay? Like acidic, soon, very soon. Yeah. So this is anaerobic, and we talk about very nice. If we were yeast cells, then we can have free flow of alcohol in our body, and without having to spend money on beer. Right, but unfortunately we're not. But it's the same idea of anaerobic fermentation, whereby we regenerate pyruvate by breaking it down to carbon dioxide and ethanol. Okay, in this process, see the NADH is regenerated. Right, we need to regenerate the NAD to continue feedback the process. Okay, glycolysis process. And finally, okay, to completely break down pyruvate into carbon dioxide and water, we need to subject pyruvate to this process called citric acid cycle, which has to be followed by electron transport and oxidative phosphorylation. Right? In this whole process, then completely 3-carbon pyruvate is broken down to the simplest, okay, simplest end product, which is your carbon dioxide. And the water is derived from the reduction of oxygen, right? Reduction of oxygen to become water. So let us look at this today. Huh? Okay, so the learning objectives, again, a lot of these pathways is about remembering the regulatory steps. We said that for glycolysis you have to remember structures. Sorry about it, but you have to remember structures, okay, for glycolysis. For TCA cycle also. And then again, we will go through how we can remember them easily without like mugging, okay, without like really just choking it in. Okay, there is a rational way of remembering. And then we look at how mitochondria is important for electron transport chain, and we derive the total energy that we can have when 1 mole of glucose is subjected all the way to aerobic glycolysis compared to anaerobic glycolysis. Right, this is your beautiful TCA cycle. Okay, remember when we talk about basics of metabolism, we said that there are a few kinds, right? There's linear, there's branch, and then there's cyclic. So this is the cyclic kind of metabolism mechanism that you see in citric acid cycle. Yeah? You don't really see it anywhere else. Yeah? Okay. So the whole idea, well, initially when we look at glycolysis, this process, glucose to pyruvate, do you notice that there's no oxygen involved? Right? There's no oxygen involved in the pathway. And where does this take place? Which part of the cell? Cytosol, right? It's only in the cytosol, right? In the cytosol. But today, when we talk about citric acid cycle, everything is happening in the mitochondria. Okay, so the compartments are completely different now. So a few questions you should have, right? So the first question. Now I have end product or intermediate in cytosol. How do I transport whatever, this product into the mitochondria for continuous reaction? So transport is a very important issue, right? And we will explain that later. So when we're looking at glycolysis, we didn't say oxygen at all, not at all. And we know that the total number of ATP derived is what? How many moles? What, per mole of glucose? 2, yeah, net is 2 moles of ATP. And then, of course, 2 NADH molecules, right? So that's why we said it's not very efficient, right? It's not very efficient with a huge 6-carbon glucose molecule. We can only derive 2 ATP after this process. Not very efficient. So the rest of the energy is actually powered by this cycle, all right? So you will see that glycolysis is actually a stage 1, okay, of what we call respiration. This whole process, you will see in textbooks, is also called respiration. Pretty much like your plant respiration. What it essentially means that we take up oxygen and we release carbon dioxide. As simple as that. Respiration means we take up oxygen at the end here, somewhere here, and then we release carbon dioxide, right? And glycolysis is part of respiration in stage 1. But at the same time, glucose is not the only molecule that can be used in respiration. We can also use amino acids and fatty acids, right? They can enter the pathway through this common intermediate called acetyl-CoA. So we have seen acetyl-CoA in the basics of metabolism, right? In activation, right? We said that the CoA can be activated by changing it to an acetyl group, a high energy compound. Besides the phosphorylated compounds, acetyl-CoA is also a high energy compound. You remember that? It's probably the only one that is not phosphorylated, okay, in the table of high energy compounds. So this is the first step. Right, and so acetyl-CoA will then, so in this part here is all mitochondria, this is cytosol. Acetyl-CoA will then enter into the citric acid cycle. And in this case, this is 2 carbon. So when you look at the cycle, also try to drill yourself on the number of carbons the compound has. Right? So you have 6 carbon to start with, became 2 molecules of 3 carbon. And then it entered the cycle as a 2 carbon compound. Okay, acetyl-CoA, and from here you will see a lot of changes in the number of carbon. So one cycle of it, the whole idea is to abstract or extract the electrons from this acetyl-CoA compound and pass it on, right? So you extract from acetyl-CoA, you get carbon dioxide and lots of electrons. Now these electrons is and the energy of this breakdown process is conserved in these electrons, and these electrons are carried, okay, by your electron carriers, NAD and FAD. Okay, that we have seen in the basics. So all the basics are coming back to link up, okay, the whole idea. So once you conserve this energy, okay, the electrons are here. So how do we use the electrons? How do we use the electrons to make more energy? So in this process then, it goes into stage 3, whereby we will look at electron transfer or electron transport and how this transport is connected to what we call oxidative phosphorylation. As opposed to oxidative phosphorylation, do you remember another kind of phosphorylation? What do you call that?
Substrate level.
Yes, substrate level phosphorylation. Very good. So in oxidative phosphorylation now, the whole idea, what is the end product? Phosphorylation means you produce ATP, right? Okay, so 2 ways of producing ATP. One is substrate level phosphorylation. Second is oxidative phosphorylation. Over here, the electron is being moved around, all right? It's being moved around across the membrane of the mitochondria and coupled to oxidative phosphorylation. So ADP, the energy, is used to power hydrolysis. The condensation, not hydrolysis, the formation of ATP from ADP and inorganic phosphate. So then, this is what we want. Okay, this is what we want to gain out of the whole process, right? Because we always say that the mitochondria is our powerhouse, energy powerhouse. And this is what we want, the ATP generated to sustain our cellular function. Okay? So these points are just what basically I have described, so don't worry, you can go back and read it. I'm not going to read it out here. So then there are three key processes that we have to look at today. Number one, the cycle itself, right? The change of carbons and how it goes back, in a circle. And then the second idea, okay, is the electron transport process. And third is the oxidative phosphorylation. So these three, okay, will make up your aerobic metabolism. And the last one is to get the idea that this citric acid cycle is a central hub in metabolism, meaning it's amphibolic. It connects the catabolism processes to the anabolism process. Right? For glycolysis, it's purely catabolism, glucose broken down, right, to a smaller compound. But you will see also later that this citric acid cycle will produce intermediates that will then go out and make other things, make amino acids, make glucose, make fatty acids, okay, in the anabolism function. So it is quite central. It links both of these two processes of metabolism together. Right? So this is basically what I have described. Okay, so the stage one now is not just glucose, but you have fatty acids and amino acids coming into the cycle. And the common intermediate is acetyl-CoA, 2 carbon. Okay, starting point, 2 carbon. And then we will look at this, okay, how it undergoes so for glycolysis specifically, we're not going to look at how fatty acids or amino acids become acetyl-CoA today, but we'll just look at how pyruvate becomes acetyl-CoA right now. Okay? So this acetyl-CoA production from glucose, okay, is catalyzed by an enzyme complex called pyruvate dehydrogenase complex. Right? You haven't quite seen this yet. This complex is a cluster of enzymes. Right? They are physically clustered together, and there are 3 enzymes in this reaction. And it is quite a special complex. You have intermediates in the vicinity of the complex. It doesn't diffuse away. It doesn't have to diffuse away and go to another site for action. It basically just happens in a very close proximity to improve the efficiency of this reaction. And then, so you have 3 enzymes, and out of 3 enzymes, you have plenty of cofactors, about 5 cofactors. And we have seen quite a number of them in the basics. And remember I said the cofactors are, a lot of them are what? Derivatives of vitamins. Alright, derivatives of vitamins. For example, CoA, do you remember the vitamin derivative? They're all vitamin Bs. Right? Okay, B5, right? Pantothenic acid. Then NAD is niacin, or B3, and FAD, B2. Okay, riboflavin. So these are all vitamins derivative. So if you look at this, this is just a recap of your CoA, coenzyme A, or the different components, the adenine diphosphate component, the vitamin component, and then the mercaptoethylamine component. So the reactive thiol group is what we want because we are going to make this compound. What do we call this bond? What do we call this bond? CS bond. What? So every time you see S, it's always thiol, right? THIO. So this is a thiol ether bond. Remember this? Thiol ether, right? Because the ether is a CO, right? Usually, right? But this is CS. So we call this a thiol ether bond, right? So this is acetyl-CoA, and this acetyl group is over here. And thiol ether linkage. Diester, right? Sorry, diester, sorry, not diester, yeah, CO. I forgot about the CO. Diester, right? It usually is COO, right? But now it's COS, diester. Thiol ether is just CS. Sorry, yeah. This is a picture, I didn't put it in, but if you go search in Google image, this is what you will see. This is what I mean by the cluster of enzymes of pyruvate dehydrogenase. And there are many units of it. You see E1, E2, E3 in close proximity, and then there will be another E1, E2, E3 combination, right? And you cluster them together under the electron microscope. This is exactly what you see, like little, what do you call it? Seed, that cereal called wheat bits. I think it's wheat bits, right? Yeah, it looks like this under the electron microscope, all right? And you draw it out, it looks like this. A whole cluster of them, and this is like a whole lot, about 60 to 100 of them in a close proximity to make the reaction really fast. Okay. So if we want to put down the reactions, yeah, pictorially. I have also added one more slide which I will give it to you. So don't panic and start copying what I'm trying to say, but I will put this slide down, okay, in IBLE so that you don't have to copy so much. But just try to understand what I will go through with you, right? Essentially, this reaction, whereby we change pyruvate to acetyl-CoA, is called an oxidative decarboxylation. Why do we call it that? Because there are a lot of oxidation reaction, right? And the end point is that carbon dioxide is removed, so decarboxylation. Okay, right? So easy to understand, right? Now, so pyruvate is quite a simple structure. You don't need to memorize, right? CH3 methyl group, right? CO carbonyl group, and a COO minus group. That's the group. Okay. So can you remember CH3 carbon 1, 2, 3, all different functional groups. So intuitively, to remove the carbon dioxide, it will be this terminal, right? This terminal group. Right? This bond will be broken, and carbon dioxide will be released. And what do you have left? This is the acetyl group, right? Acetyl group. Now, the acetyl group is being transferred to the first enzyme in the complex called pyruvate dehydrogenase, and this is the slowest step in the reaction. Okay, this is the slowest step in the reaction, is the rate determining step. The acetyl group, okay, will combine with one of the functional groups or the cofactors, right? This is the cofactor in the enzyme, TPP, which is thiamine pyrophosphate, to form what we call hydroxyethyl TPP. This is the intermediate. CH3, instead of CO, you have an OH here. Okay? So then this reaction, okay, will somehow proceed, right? Now the end product is to make acetyl-CoA. To combine, we have to combine this acetyl group to a CoA. CoA is always swimming around in the body, right? So CoA will combine with it to give you acetyl-CoA. This is what is desired. And throughout the process, there will be a pair of electrons that needs to be transferred out. Okay, because essentially we are moving just the acetyl group. The pair of electrons will end up here in NADH, right? Okay, reduction process. NAD is an oxidizing agent. When we say we have a reduction process, it means it has that means it has gain of hydrogen or gain of electrons. So that pair of electrons is being gained by NAD to give you NADH, and NADH will then be the electrons will then be used, okay, to be transferred to oxygen, and the end product will be water. Right? This is the last step. Right? Okay. So then in the middle of the process, what we're really essentially looking at is the movement of the electrons and the acetyl group. So the first step is the slowest step. You have the acetyl group over here, right? And the acetyl group is being passed to E2, the E2 enzyme. What is special about this E2 enzyme is that it has what we call a lipoyl functional group over here. You see this lipoyl functional group over here. So what do you call this? Thiol, right? This is the thiol group, THIOL, SH thiol group. And what do you call this? This is the disulfide bond. Okay, this is classical. We see this also in our amino acid. Which amino acid is that? Cysteine, yes. Cysteine can make a disulfide bond like in your curly hair, right? Remember, I don't know if Dr. Ho mentioned about curly hair, but when you do perming of the hair, what you're essentially doing, right, is you're oxidizing your cysteine molecules in your keratin to make them form disulfide bond. That's why the curls hold. Right? Okay? So basically this is what is in E2, the lipoyl functional group, which has two thiol groups. Now this two thiol group becomes disulfide, okay, oxidized to get disulfide group. And then this disulfide group can then be used to carry the acetyl group over here. Right? So this is a series of passing on the acetyl group. Right? You pass the parcel, yeah? So once it is being passed, okay, to this E2 enzyme, what we call a trans-esterification process will occur here. That means the whole ester group, right, is being passed on to the CoA functional group. Right? Trans-esterification process in number 3. This whole functional group will be passed on to CoA to give you acetyl-CoA. Alright, in return, the enzyme is regenerated. Right? So again, you see that the H here, which is the pair of electrons, is not addressed. We don't see the H here. Right? We don't see the H here. So it must be floating somewhere around. Correct? Yeah? So the H here is floating around here, and you must regenerate. In order to regenerate this lipoyl group for continual function, the other reactions over here, right, basically serves to regenerate this by taking away the electron, the pair of electrons. And so you will see FAD again in a whole series of oxidation reduction processes, then move the electrons finally to NAD+, okay, to give us NADH. Okay? So when we see the electron transport chain later on, again, the whole concept again is to pass on the electrons. You must move the electrons. And how do we move electrons? We change the oxidation reduction rate of these electron carriers to help us move it to the final acceptor. Right? We said this is the final electron acceptor, which is oxygen. Okay? That's the main idea. This is just a summary of the different prostatic groups and coenzymes involved in the whole process. Right? Okay, so it's linked to this description which I will give you. Yeah? Right, so that step is a preparation step. Acetyl-CoA is not in the cycle yet. Right? It's just a preparation step for us to get it from pyruvate, but already it is happening in the mitochondria. Okay, so from this top here onwards, everything is in mitochondria. You have to remember that. Okay? So now, acetyl-CoA will then enter the citric acid. There are so many names for citric acid cycle, right? Tricarboxylic acid cycle, citric acid cycle, Krebs cycle, right? And then next week, the next lecture, you'll probably see, next week you will see another thing called Krebs bicycle. You know? So many things. Right? There's a very interesting one, Krebs bicycle. Okay? You can imagine it to have 2 circles there. Yeah? Okay? So, but for us, the Krebs cycle is just 1 circle, right? 1 cycle. Again, numbers. How many steps do we have in glycolysis? 10, correct. 10 steps in glycolysis. So how many steps do we have in citric acid cycle? 8. Right? This is not going to be an MCQ question. It's too easy. Okay? So we have 8 reaction steps and all in the mitochondria. Okay? In the mitochondria, let me stress. Yeah? So how do we get, okay, so in this reaction, we have acetyl-CoA combining with what we call oxaloacetate. This is 4 carbon. Okay, 2 carbon. And it enters the cycle and becomes a 6 carbon straight away. How does this happen? Okay, because it combines with oxaloacetate, which is the 4 carbon compound. Okay? Yeah, it combines with this to give us a 6 carbon. Now, then the carbon doesn't really change until we remove it in the form of a carbon dioxide. Right? Remember again, our end product is carbon dioxide and water. So now carbon dioxide removed. So from 6 you reduce it to 5, right? And another carbon dioxide molecule removed, you get 4 carbon, all right? And from henceforth, the carbon doesn't change, all right? You see, from 6, once the carbon dioxide is removed, okay, C4 all the way, doesn't change anymore, okay? Doesn't change anymore. So what do we get out of this whole cycle? Okay, acetyl-CoA, C2 becomes C6, right? And oxaloacetate is being used. One oxaloacetate is being used, but it is regenerated at the end of it. So there's no net loss of oxaloacetate. That's why it's a cycle. No net loss. There's no net loss of oxaloacetate, right? But at the end of it, we extract 2 moles of carbon dioxide from it. Okay, we extract 2 moles of carbon dioxide reaction on it, and we didn't show, right? And we didn't show. We didn't show in the reaction that out of it, as long as you see a dehydrogenase reaction or carbon dioxide extracted, we have electrons being passed on to NADH. Right? So we also need to know that how many NADH are being produced and FADH. Well, it's actually summarized in here. Let me just go through this. Yeah? One cycle results in 2 moles of carbon dioxide released, 3 moles of NAD reduced to NADH, 1 mole of FAD reduced to FADH, and 1 mole of GTP or ATP produced. Okay, we will need this information to calculate the total number of ATP derived from the whole cycle. Alright, okay? So let's go back. This is what I have described. Let's go back to this cycle and look at it in detail. Okay, we're going to look at the reactions in there. I'm not going to cover all the reactions because not all the reactions are critical. I'm just going to point out certain points, okay, about this reaction. Okay, so again, my objective here is to help you to remember the structures and the movement of the structures. Okay. Okay, so now let's start with acetyl-CoA, C2 to C6. This reaction, this first reaction is highly exergonic. Alright, that means it is a forward reaction, doesn't ever go back. Do you see like all the other reactions, almost all will have a double arrow. But this reaction is a single arrow, it never goes back. You have to make it go forward, if not this cycle wouldn't move. Okay, this cycle would be like flipping flop, you know. So you have to make it work in a clockwise fashion, and so you must make sure that this reaction is highly exergonic. And so the reactants is found in very low, okay, as we have mentioned, the basics of metabolism. In a highly exergonic reaction, the enzymes are usually far from equilibrium. We have this concept, right, that the enzymes are far from equilibrium. That means that the substrates are always in very low concentration. Whatever you have, it becomes product. Okay? Whatever you have goes to become product straight away. And the enzyme over here, okay, so this reaction, you would think that it will be highly endergonic. And fortunately, we have a high energy compound, acetyl-CoA, to power the reaction. Therefore, the reaction goes forward, okay, in a highly exergonic fashion. So high energy compound will then power citric synthase towards this reaction. Okay? So then you can say, you can kind of guess already that this is one of the major regulatory points of the cycle. Okay, this is the major regulatory point of the cycle, citric synthase. Okay, pretty much like glycolysis, we identified the regulatory enzymes, right? A few, about 3 of them. Okay, in the Krebs cycle, also we have about 3 of them. And this process here is not quite regulatory. It is just the dehydration and rehydration of the molecule to move, right? To prepare the carbon dioxide to be removed. Now, the one shaded in pink is the acetyl-CoA, is the carbon atoms from acetyl-CoA, right? We will explain why we highlight in pink, okay? So you see over here, okay, once it combines with the oxaloacetate, it becomes, you have 3 COO minus group over here, 1, 2, 3, okay, and it's called citric. Right? Citric has 3 COO minus group and 1 hydroxyl group on this side. Right? So through successive hydration and rehydration, because this is a cis conformation, the OH group can be moved from here to here between the 2 carbon atoms. You see, essentially you're moving the OH group to this carbon atom. Very typical of a cis kind of compound, right? Some kind of symmetry to it. So once you move it over here, it can then form what we call isocitrate, right? So isocitrate, and it goes through the first decarboxylation reaction to get this carbon dioxide removed, right? And it is facilitated by isocitrate dehydrogenase, again a single arrow. This is the second point of regulation, right? Now dehydrogenase, ring a bell? The word D, the enzyme dehydrogenase. Remember I said that whenever you see the enzyme called dehydrogenase, it will always be coupled with the electron carriers. And the electron carriers, we only have 2, right? NAD and FAD, right? So not like a lot to remember. So the dehydrogenase is coupled, okay? This enzyme always couples this reaction with the reduction of NAD. So in the end, not just carbon dioxide is produced, but you have electrons being passed to NAD to give you NADH. Similarly, so the end product is called alpha glutarate. And so we remove 1 carbon, 1 CO2 group, and then remove again another CO2 group. Alright? Okay, and this is the alpha ketoglutarate dehydrogenase. So we always name it with the substrate. Over here is isocitrate is the substrate, dehydrogenase. Over here, alpha ketoglutarate is the substrate, so alpha ketoglutarate dehydrogenase. Okay, and with CoH coming in, you have CoA, or CoA is usually written with an SH group to remind you that it has an active thiol group there, but it's actually a CoA enzyme. So now CoA comes in, okay, and then you have carbon dioxide being removed, again coupled with NADH. Gives you another mole of NADH over here to give you what we call succinyl-CoA. Right? So this alpha ketoglutarate dehydrogenase is a complex. This complex is the same as the pyruvate dehydrogenase complex. The same idea, the same concept, 3 enzymes together, lots of cofactors. Right? So the same structure. Right? And over here, succinyl-CoA, doesn't it kind of ring a bell? Is it the same as acetyl-CoA? See the CoA over here, CSCO. Right? So what's the difference? Acetyl-CoA, we have only this, right? CH2CO. But succinyl-CoA, we have 1, 2, 3. We have 3 carbon, 1, 2, 3. Right? Okay, acetyl-CoA is a CH3, sorry, a methyl group, but succinyl-CoA has 3 carbon with a COO minus. Right? And anything CoA is a high energy compound. Right? Right? It's activated. The CoA gives it this activation or high energy that it has. So this is also a high energy compound. And what do we use high energy compound for? What do we use high energy compound for? For ATP production. Right? So the concepts is running through it again. So in this case, the next step then is to use it to make ATP or GTP depending on the isoform of this enzyme called succinyl-CoA synthetase. Alright, so that's why you see a bracket, GTP or ATP. Doesn't matter. GTP can also be used or converted to ATP as an energy source. Doesn't matter, right? So depending on the isoform in the cells, you can either have GTP or ATP. It is still counted as energy. But in this step, we call it a substrate level phosphorylation. Okay, remember like glycolysis, we also have substrate level phosphorylation whereby the phosphate group is passed on to ADP directly without the involvement of electron transport, without the involvement of oxygen, right, to make ATP. That's why we call substrate level. So over here, we make 1 mole of ATP or GTP by passing on the inorganic phosphate directly, right, to the ATP or GTP, right? Okay, and it's facilitated by a high energy compound, in this case succinyl-CoA. and then the CoA gets reduced, and you get succinate. See, succinate is a completely symmetrical compound. COO minus, CH2, CH2, CO minus. It's a symmetrical compound. Okay, symmetrical compound. And the subsequent steps then is to prepare. Okay, not too difficult, but this step will then prepare the molecule to become oxaloacetate to return the action back again in a cyclical fashion. Alright, so from a symmetrical compound, okay, we will end up with oxaloacetate. And the difference is this CH2 here becomes a carbonyl group. Alright, difference is it becomes a carbonyl group. Right, yeah, and then when this is regenerated, it goes again and again and again. Do you notice that the first two molecules of carbon dioxide remove the carbon does not come from acetyl-CoA? Okay, in the first cycle, the carbons from acetyl-CoA is not removed. Only in the second cycle, then it gets removed. Right? Did you see? It is being preserved. Okay, this pink bar here, right, untouched until the next cycle, then it's being removed. All right, so this is something special, okay, about the sequence of the events. Yeah, see the pink color here? Yeah, it is not removed. Only in the next cycle. Okay, so again, for the pathways, we have to look at the regulation of the cycle. And in this case, we have regulation from outside and from within. Outside and within. From outside the cycle, that's simple enough. There's only one reaction, isn't it? There's only one reaction which involves the pyruvate dehydrogenase complex to make the acetyl-CoA. Right? And from inside, we have three major enzymes with the single arrow, right? Very far from equilibrium, highly exergonic. And I highlighted here citric synthase, isocitrate dehydrogenase, and alpha-ketoglutarate dehydrogenase complex. These are the major control points. Okay, so looking at this, so glycolysis, we talked about the inhibition, and then we saw that the inhibitors usually are ATPs. Right? ATPs. Why do we use ATP to inhibit? Because ATP is an indication of what? That your fuel is enough. Your fuel supply is enough. Right? So the same thing you will notice, yeah, that ATP NADH is the inhibitor for this process. So if you look at pyruvate dehydrogenase, there are two forms of regulation. Allosteric regulation is what we have seen commonly right now. Okay, you see it very frequently. But on top of that, pyruvate dehydrogenase has an extra regulation called covalent modification of the enzyme. And this is a post-translational kind of modification. Okay, so nothing rocket science about it. This about what I want you to gather from this chart. Okay, it's quite simple, just to recognize the inhibitors and activators. So product inhibition, right, is straightforward. Remember I said that the end point is to get acetyl-CoA, right? So this is the product. And the other product is NADH, because the electrons are not being passed on to acetyl-CoA. The electrons from this compound is being passed on to NADH. Right, so these are our two products. And this will follow our standard allosteric inhibition. Right, so acetyl-CoA, excess of acetyl-CoA and or NADH will then slow down these enzymes. Okay, the whole complex works in tandem, right? And this is the slower step. So it will have a lot of impact on E1, on the rate of action of E1. On top of that, for the pyruvate dehydrogenase enzyme, you have covalent modification, whereby it works slightly different now. It doesn't look quite like allosteric regulation. You have the involvement of extra enzymes, okay, extra enzymes. And these enzymes the covalent modification uses phosphate group to inactivate the E1 enzyme. So if you look over here, these activators are not allosteric, they are not activators for E1, they are activators for pyruvate dehydrogenase kinase. See the color coding? Alright, so the red color here, yeah, this side here, these are activators for pyruvate dehydrogenase kinase. Meaning, if you have the same, you see, don't you memorize? Same thing, acetyl-CoA and NADH, if you have too much of it, and feedback inhibition is not enough to control the enzyme, then let's do a covalent modification. How do we do that? We activate this thing called kinase. And what does kinase do? What does kinase enzymes do? Add or remove phosphate groups. Add. Okay, standard kinase terminology means this enzyme will add a phosphate group to whichever enzyme it's working on. Right, so this kinase adds on a phosphate group into E1 over here, and this inactivates the enzyme. Okay, so how does it inactivate it? It has to be with structural conformation changes, right? Right, so once it adds this, this phosphate group, the enzyme inactivates, then the whole process stalls. And on the other hand, if you have inhibitors of this enzyme, okay? So if you have raw products, these are all raw, most of them are raw products, pyruvate, ADP, and these are cofactors for the enzymes. If you have too much of the raw product and too little of the substrate, if you have a lot of the substrate, then this enzyme will be inhibited, all right? Instead, the phosphatase enzyme will be activated. So if it is inactivated over here by a phosphate group, then you need to remove this phosphate group. This phosphatase enzyme will remove this phosphate group and make E1 active again. Alright, so I don't know if you have seen this kind of regulation up to this point, but this is different from the usual product inhibition kind of activation. Okay? So with the help of extra enzymes, you inactivate it. The whole idea is to change the conformation of your enzyme in question. Okay? So basically this is to describe what I have said. And under this situation, especially when you have high glucose, later on you will notice that when you have a lot of glucose in the blood, right, insulin hormone will be mobilized, right, and it can activate PD phosphatase. Right, to activate the enzyme to remove the glucose. Right? You cannot have a high glucose circulating in the blood. That causes a lot of damage. You must mobilize the glucose and make them leave, okay, the bloodstream to go into your tissues. And so insulin's job is to do that. Right? Once it goes to the tissues, into the cytosol, mitochondria, then it will be broken down. So for the other three enzymes, nothing too stressful about it. Yeah, I just want you to, again, it's very simple. The allosteric inhibitors, the inhibitors are working in an allosteric fashion. We have seen it for the phosphofructokinase, hexokinase, and the pyruvate kinase, right? And these are all the products. And did you notice that citric? What does citric do to glycolysis? Do you remember? Do you remember seeing citric somewhere in the last lecture? What does it inhibit? What does citric inhibit? Which enzyme? Which is the major regulatory point in glycolysis? Yeah, phosphofructokinase. There are three, right? So the second one called phosphofructokinase is the major point, and then citric is an inhibitor. So why is this happening? Okay, don't you see now that the glycolysis and the Krebs cycle has to be linked up? The control has to be precise. Why? Because you should only have so much glucose that the citric acid cycle can manage. Okay, so there is a tight control between the two processes. If the cycle cannot manage, then the cycle must have kind of a feedback to the top process of glycolysis, the earlier on process, to slow down, okay, the fuel production. So citrate is one of the special ones that is being fed back earlier on to glycolysis to slow down the process. Okay, so all these connections you have after you see all the pathways, you kind of have to make all the lateral connections between the different pathways. Yeah, so that's the fun part. So the citrate will go back to glycolysis. Yeah, and over here these are normal ATP, succinyl-CoA, NADH. Okay, so ATP and NADH you will see in most of them because they're the products. Succinyl-CoA is high energy product. It will definitely move the reaction forward. Therefore, it makes sense to have it as an inhibitor. Alright? So the ADP is the kind of like the raw product. Okay, this will be an activator. So in the body, this balance, this ratio is tightly controlled. ATP to ADP. Okay, so if the ratio is pushed towards having more ADP, alright, then it is time to signal the cycle to go into action. So these three concepts of inhibition is seen throughout the three enzymes: substrate availability, okay, product inhibition, allosteric regulation. Right, so it's almost quite a boring MCQ question to ask whether which one is an inhibitor, because you will see even for isocitrate dehydrogenase you get ATP and NADH, alpha ketoglutarate you get ATP and NADH. Right, and activator you see ADP, NAD. So the idea, sometimes you see calcium and magnesium, it is because they are the cofactors for the enzymes, especially for dehydrogenases, right? So that's why they are there. Okay? Right? Okay. So the regulation of this TCA cycle is a little bit quite straightforward. Quite straightforward. Again, the same concept: product inhibition, allosteric regulation. The other thing, remember I said that we have to recognize that TCA cycle is central. It's the highlight of metabolism, right? Because it links both anabolism and catabolism together. Did you see? If you see this figure over here, we haven't covered all these things, right? The next lecture is about fatty acids. Then we will look at amino acids. There's no nucleotides here, so nucleotide is out here, okay? Out of here. But do you see how they are linked? Red means anabolism. Green means catabolism. Okay? So meaning that this citrate product can be used to make fatty acids, can be used to make cholesterol. Malate can be used to make glucose, and succinyl-CoA can be used to make porphyrins. Porphyrins is in your hemoglobin in your blood, and your cytochrome C is in your electron transport. Alpha ketoglutarate can be taken out to make amino acids. Right? So this connection between anabolism and catabolism is central to this cycle. We have to recognize, and the term used is called amphibolic. Right? Because amphi means both, yeah, both loving. Right? So you have catabolic and anabolic processes. And the catabolic processes is just what we have gone through. The carbohydrates, fatty acids, and amino acids enter the cycle via the acetyl-CoA determinant. And over here, the anabolic process gets a little bit more complicated. Right? Glucose synthesis, we did not go through. It's a process called gluconeogenesis. There's no time to go through it. But recognize that this is a raw product for making glucose. Okay? And then citrate is used for fatty acid synthesis, and alpha ketoglutarate is an oxaloacetate used for amino acids. Alright? So if you wonder sometimes, how did we ever even start with amino acids? Where did they come from? Right? It is from, again, back to glucose. You can make, the whole idea is that glucose has so many carbon, we call it the carbon-hydrogen backbone, right? And this carbon backbone will then form the basis for your amino acids. Okay? Right. So we still have not addressed this electron issue. If you go back to the cycle, okay, glycolysis, we have 2 moles of ATP, and citric acid cycle, we only have 1 mole of ATP directly. So it's still not good enough, right? It's still not good enough. You mean that it's going to power us our whole daily function, our cellular functions? So where is the other energy? Remember the first law of thermodynamics. Nothing gets destroyed or nothing gets created. The system in the universe stays the same. So from carbohydrate, from the glucose, I should be able to extract all the energy available, right? And so the remaining part of it then goes to the function of what we call electron carriers. And we have 2 electron carriers, NAD and FAD. Right? Let us count how many electron pairs are released for 1 mole of glucose, right from the beginning. So if you remember for your glycolysis cycle, we have 2 NADH. We have preparatory stage, right? We said this is an investment, energy investment. We use up 2 ATP, we use up NADH. And then we have the payoff stage whereby we make ATP, right? So the profits from the investment. So over here, the glycolysis, we have net 2 NADH produced. Now this is in cytosol, okay? This is in cytosol, 2 NADH produced. And then over here, just now we mentioned while making acetyl-CoA, we also have 2 NADH. So we put a 2 there, right? Do you remember why we put a 2 there? Because glucose is C6, pyruvate is C3. Every mole of glucose, we get 2 moles of pyruvate. So now from here, everything we add 2, yeah? So NADH, so in the acetyl-CoA section, we have 2 NADH molecules, okay? And then throughout this process, remember we said the dehydrogenases. We have 3 dehydrogenases, and over here, the succinate dehydrogenase is a bit special. It doesn't use NAD, it uses FAD instead, okay? Why? Because this enzyme is located on the mitochondrial inner membrane. It's not swimming around in the matrix. Okay, all the others are in the mitochondrial matrix. But this one is on the membrane, and it is very closely linked up to the electron transport complexes. And it uses FAD. That's the difference. Okay? It is not in the matrix. So, and then there's another one here at the end. So, do we have 10? So, 2, 4, 6, 8, 10. Okay? So, 1 cycle, 1 mole of glucose, we have 10 NADH, and we have 2 FADH2. Okay? So, what do we do? Now, these electron carriers are all reduced. Every one of them are carrying a pair of electrons. So that's why we say there's 12 electron pairs, right? 12 electron pairs. Where do the electrons go? Finally, they have to end up with oxygen, right? They have to go to oxygen. Remember our final electron acceptor. So how do they travel all the way to oxygen? Okay. So let us take a look at the structure of the mitochondria first. I keep stressing that the process is in mitochondria. Why? Because this mitochondria is quite tricky. Yeah? There is an outer membrane and an inner membrane of the mitochondria. Alright? So the outer membrane is freely permeable to small molecules and ions. Protons can go in, small molecules can go in and come out. Now, then the problem now comes with the inner membrane. Inner membrane is impermeable to most small molecules and ions, including protons. Can you imagine? Protons is so tiny and couldn't even go through this inner membrane. Right? Okay? So what you have now is the inside here, the matrix of the mitochondria. Right? The matrix has all your enzymes. You see? Pyruvate dehydrogenase complex, citric acid cycle enzymes, fatty acid enzymes, amino acid enzymes. DNA, right? We talk about your mother's DNA, right? Inside here from your mother. And then all the other products, all the other enzymes and intermediates. They will not be able to come out of the matrix. All these things stays in the matrix. Okay? Right? So how do we, then there's some problem here already, right? Because Just now we saw that we have NADH produced in the cytosol. How do we make it move into, make them move into the membrane or into the matrix? There's no way. Right? So what the mitochondria does is to have specialized transporters. Right? This sounds like the movie Matrix, but anyway, so it has specialized transporters on the inner membrane, right? Which are channels, which are pores that can open, gates that can open to let certain things in, right? So over this, on the inner membrane, you have respiratory complexes. These are our electron carriers. They are proteins, so these are all proteins. And you have your ATP synthase, okay, we'll describe this, and other membrane transporters. Okay? So you can, one of the transporters that we need, which one do we need? Pyruvate. Alright? Pyruvate is made in the cytosol. We need it to be converted to acetyl-CoA, right? And our pyruvate dehydrogenase is inside the matrix. So we definitely need a pyruvate transporter in this membrane. And some others. Okay? So the classical anatomy of a mitochondria, you have convolutions called cristae, right? Gives it a very high surface area. And this high surface area will give you about 10,000 sets of the electron transfer systems. So this is really a powerhouse, right? This is the powerhouse. It has about 10,000 sets of respiratory chains, ATP synthase molecules to make ATP for you. Alright, so that's why mitochondrial diseases are devastating. Okay? Okay, so now get this anatomy in your head. Nothing can go in without the help of transporters. Nothing can come out with the help of transporters. And all the major enzymes are trapped inside this matrix. So again, back to